So, good morning, everyone. I am Dr. Pradeep Sharma, the General Secretary of Biofootprints, a society for promotion of science. And I, on the behalf of the society, I welcome you all here. Today, we have amongst us a very renowned scientist, Dr. Peter Male. Dr. Peter is the head of Laboratory of Ligand Engineering at the Institute of Biotechnology, Czech Academy of Sciences, Czech Republic. He graduated from the Institute of Molecular Genetics of Czech Academy of Sciences in Prague. He did his postdoc from HHMI, University of Michigan Medical School, USA, in the laboratory of Professor John, where he published several papers related to in vivo role of mammalian glycosyltransferases. He has been the editor of several scientific journals. His current focus is the development of combinatorial protein libraries derived from small protein domain scaffolds and the construction of novel high affinity protein binders with therapeutic and diagnostic potential. So now I request the president of Biofootprints, Professor Sujata Sharma, to welcome our speaker with a bouquet of flowers. The photo.
Photo. Video photo.
Thank you very much. Thank you very much. Very nice invitation and introducing her. Thank you.
Now I request Professor T.P. Singh. He is a third distinguished professor. He has been here for 4 decades to say a few words.
I can only appreciate good things. Good morning. It's a great pleasure to have you here. I'm very familiar, not very familiar now, since many, many decades back I have been to Prague in German. It's a very fantastic country, very wonderful country. And I think one of the frontline countries in Europe. And their science, even now, is outstanding. And it's a pleasure. I think the kind of work you are doing, we have great interest in this. Maybe we should think of some kind of collaboration in future. We have this DST Czech Republic collaboration. And this is our great interest, what you are doing regarding protein engineering and high affinity ligands and drug discovery is very, very exciting. So we are very happy. I'm very happy. Thank you very much for coming. All the best.
Now I request Dr. Peter to come and deliver his talk.
Okay. Thank you very much. So you can start the presentation. Just yeah. Okay, let's okay. This icon, right? Yes. Okay, so thank you very much again for a very nice invitation. I'm really happy to have a chance to talk to you. And because this is a very special meeting, so I don't know how much time you have, but I thought that it will be nice to present some of our projects which are related to some kind of application in medical sciences. So, because this is all Indian Institute of Medical Sciences, so I thought it would be nice to select from our project. We have several running projects. Those, who could be interesting for you in some relation, how we can generate specific protein binders that can be useful for a wide range of biomedical applications. So, let's start with the first slide. Okay. This is the okay. So this is our research center, which is not high but pretty large in area. This was established and opened in 2015 year. So we stayed there 7th year, and this was supported by European Union supporting grants, funding via Brussels money to support some regions and some areas. And our location is called Westec, which is the pretty small town on the border with our capital Prague. So we are not in Prague, we are just 1 kilometer behind the Prague. So this is a region called Central Bohemia. It's like, Uttar Pradesh around Delhi. Okay, so it's Central Bohemia region around Prague. Okay, so we have many students from Prague, also international recognized, and this research center was supported by Charles University, which is the most famous university in our country, and it is actually second oldest in Europe. Okay, it was established in 1346 by Karel Charles IV, Caesar at the time, Czech origin from France. He established that university, so it's the second, the oldest one in Europe. The oldest one is from Italy, Lausanne. Okay, so just
So this was in 1300, you said.
1346, I would was
Really old.
established, pretty old, very traditional one. Got many faculties, it's scattered through the whole capital, even from other countries. And we share, Academy of Sciences is an institution which is far from university, which is like a special system in our country, which differs from most European countries, where have institutes related to universities. So Czech Academy of Sciences is separated, but we share, leadership and supervisors, students from university. So they are working in our laboratories, and we work as a teacher, supervisors, of these students for Charles University. So there are locations of several institutes, and Institute of Biotechnology is the major one, sitting in this area. And I made the title of my today presentation like a more general one, like development of small protein binders as useful tools for biomedical applications. So let's talk that. My laboratory is called Laboratory of Light Engineering. So our mission is to develop techniques and artificial proteins with some function we want to design. So basically, what we want to do is increase specificity of proteins or generate specificity and high affinity, so improve something or generate some novel proteins. Those can be used for in vivo diagnostic, for instance, as a capture proteins with high affinity on the layer of biosensors, for instance, or you can use them to develop some innovative ELISA kits to test the presence of some important molecules in body fluids for patients, let's say. The other direction is focused on some immunomodulatory research because we would like to affect some pro-inflammatory processes by generating inhibitors of some signaling molecules. So something similar like people are developing with antibodies or antibody fragments. So we are doing something similar using small protein binders. So typically what we want is to develop some inhibitor of function, so something neutralizing ligand, for instance, to block cytokine or its receptor, stop signaling to function. Okay, so I selected one typical something for the diagnostic research we have. And the last part of my presentation will be focused on something which is quite new and quite interesting now. This is our original approach, how to develop vaccine antigens. So this will be my part. So I don't know, how much time you have, but if you think I'm too long, just stop me. Okay, okay, okay. So let's start with the first part of my presentation. So just to summarize, you're from Department of Biophysics, so you are familiar with this, but this is like an overview, protein engineering. What does it mean? So what we use is directed evolution of proteins. And this actually is the method once you develop new protein binders. And you can use our selection from highly complex combinatorial libraries, which we assemble, and then you have molecular target, and you try to identify protein binders specific for the molecular target by screening from many variants in the library. The other way is to use in silico approach, to design something and then to try to produce it, verify function, and then correct it back and modify it. We mostly use that screening from combinatorial libraries, which is our domain. And regarding in silico modeling, bioinformatic analysis for this part, I actually use the other laboratory, which is the Laboratory of Structural Bioinformatics of Proteins, led by Jerry Cherny. He's a head of this group, and so I share with him some, these activity in terms of prediction of binding sites of ligands and suggestion of improving mutations. So doing this analysis. So we are mostly oriented on the experimental work, and this in silico work is done in collaboration with the other group. So we have like a library. We actually design our own libraries for this purpose. I will talk it and use a method which is called ribosome display. We establish in our lab. This is a method published early in 2002 by researchers from Switzerland, Dr. Plichtun, who's actually established this technique, published, and we just use this method for identification of our protein variants. Okay, so we are also building some homology models, for some purpose, so doing this part. Okay, so first scaffold we established in our laboratory about 10 years ago was the albumin binding domain scaffold, which is a very small 3-helix bundle, which is very small domain derived from streptococcal protein G. And this has 3 helices, and actually function of this domain, which is shown here, serum albumin is bind to, this is the domain of streptococcal protein G, which is responsible for binding to serum albumin. And we used this as our master scaffold for generation of highly complex combinatorial libraries. This is only 46 amino acids. Okay, it's a very small one. And we use it and perform some in silico analysis of mutable residues, and then we designed 11 randomized positions in the scaffold, which can be used for making highly complex combinatorial library with the theoretical library size is 10 to 14 variants. Okay, so this is what we did. Actually, we mutated these amino acids position and by randomization, and which is shown here. So we used one of 3 surfaces to be randomized. And we selected we call that surface B, because this B surface is responsible for high affinity, natural high affinity of this albumin binding domain to serum albumin. So there are critical residues located here for binding to serum albumin. And by our concept of our library, we actually destroyed these critical binding sites and substituted them for the newly engineered affinity.
Yeah.
So selected these ones
So
exactly.
when you mutate 2 objectives to enhance affinity or you to generate a structure. So when you mutate, you don't lose the structure of helical structure.
No, we just did it alanine scanning first to be sure, okay, that stability protein will not be disturbed, and identify those residues which can be mutated, still keeping 3-helix bundle structure. Okay, so this was like a in silico, prediction to mutate exactly this position, and then you can randomize in
So
alteration.
you did one by one. You mutated one by one, or
No.
residues?
We are using synthetic approach. So we have like a synthetic DNA primers or adapters, what we have, and those are randomized during synthesis. Okay, so you synthesize molecules of DNA, and each template, I mean, like a molecule, you have a different combination because they are during synthesis, they use the mixture of nucleotides. So they are randomized randomly. So you have a mixture of molecules for synthesis. I will show you then in detail how we do it. This is the only like a principle that we identify these mutable residues doesn't disturbing this three helical structure or are predicted not to disturb. You cannot be sure, of course, once you're doing new molecules that something will be different, but in prediction, this is the most stable way how to generate library. Okay, so we actually diminished original binding to BSA and then randomize it, and so we generate on the same surface new set of residues, allowing you to actually engineer new affinity specifically for the chosen target. Okay, so now we decided to work on another concept of scaffold protein, and this is we use the human myomazine 1 gene, which is the gene which is used to work here in sarcomere, moving these binding also myosin and titin, and this protein has a 13 these domains, and we selected one of them, which is shown here in the blue color. This is domain 10 on human myomazine 10, and it generated as a scaffold based on beta sheet structure mostly and some loops to generate or design another concept for another scaffold, and which is shown here. You have loops here like in antibodies. Okay, so you can randomize these amino acid residues in the loop region. Then it's stabilized by 7 antiparallel beta sheets with some supporting helix there. And we crystallize this structure, put it to PDB with the code. It's not important to show you that code, but I'm just saying that we measure stability of proteins, which was 72 centigrade. In the original, we call it wild type, and we call that design biomedical, which is our name, how we call that scaffold. And yeah, we purified it. Its size is 111 amino acids. There's a PI6, 13 kilodaltons. Yeah, and we purified it, tested it, analyzed it, measured stability, crystallized it. And then we used to make design. Okay, so this is the myomedin scaffold randomized 12 position in this case. So theoretical library complexity is even higher than there. Okay, and we use a TRIM library, which is the way how to synthesize the DNA pieces to assemble library. And TRIM means that you actually assemble 3 nucleotide codons. So you don't randomize particle bases, which is the case over here, which is called the NNK, because you randomize nucleotides. So you can even generate some stop codons or proteins having cysteines. Here we eliminated cysteine residue because we don't want to have such complication with making disulfide bonds in these. So we actually randomized only 19 amino acid types of residues eliminating cysteine. And during the synthesis, this was synthesized by codon combination. Okay, so it's codon, directed synthesis. So it's actually better because you avoid problems with the stop codon, randomization of one of three stop codons, and also we eliminate cysteine. Okay, so this is how we assemble. There is several steps. You have parts of adapters and proteins. So there is several step of PCR transcription to assemble a library, because you have to you install ribosome binding sites, because if you use a ribosome display, so you want to do it in cell-free in vivo transcription and translation. So you have to have all these parts done. Okay, and we also use some parts of this stabilizing protein here. So this is just to understand how we do it. So we ordered from the synthetic company from Germany pieces of these with these randomized loops here, and then we use this DNA to combine and transcribe and finalize. So, and actually we have two concepts of myomedin library. We call that loop library and beta sheet type. Okay. In here we randomize 12 residues in 3 loops. Here we randomize the flat beta sheet surface here with here one loop, but mostly here. And this was also pre-designed by in silico docking, predictions, running scanning, and this just to be sure that we can use it not to disrupt the general fold. Okay, so this is to actually work with this C terminals, and this allows produce area strains which have a beer A patients coding protein case and in biotinylate this protein bacteria do, because this is the orange biotinylate. So you know where the biotinylate protein bound to, and trick how to produce these proteins to be detectable. Okay, so also we collaborated with a group of people in A, and they are in real life working with patients. There is hospital in hospital, and they have also. So we asked the question: wild type really in terms of it being a non-specific kind. So sent then the myomedin wild type. They use it for radioactive labeling, gallium. Then they put the myomedin wild type radioactive label in place, and then they're doing 5 minutes. Hence, your point was to verify if there is non mouse organs. And what we can see here is where you because events leather whole serious population urine-related organs hearing. So this is actually a very good result. Try to generate specific features, for instance, for cancer, to be sure that myomedine inside the body doesn't cause any false signals. Original called cell type randomized doesn't bring any kind of substantial non-specificity. Okay, so we used ribosome display. Is for cell. So we actually ordered it for transcript translation kit and having these ribosomal units. What you do is that it take up things for combinatorial library, which is type from A to mRNA, then packed to the ribosomal complexes, can pull these complexes by via interaction with the targeted protein, which is the protein of choice. So we, for instance, say we need this against some medical important protein, so we know which protein we have as a target. This could be put on the plastic dish, 6-8, but you can also use particles like magnetic particles and immobilize, and they are like a target for this mixture of ribosome complexes. And point is, in our library, once we design, we actually eliminate codon because you don't have codon. Subunits part, they are still together. This allows you to really pull all the complexes from targets and push whatever else is not binding or with a low affinity. So you can really select only those with a substantial affinity. This is the way how you can identify these proteins. And once you identify them by binding, well, you are interested in messenger RNA, which is coding your proteins. So you put it to stimulate degradation of, let's say, to do reverse transcription, and you go to one round of, okay? You can do it several times. Repeat this. Typically, we are doing sometimes 5. You can increase even 7. And point is of this method is that in every round of ribosomal, selection, you can increase stringency. You put lower amount of your protein target. At the same time, you can increase washing cycles, and you can even increase detergent. So you make interactions. So by controlling these steps, you can select medium affinity binders, or you go to the really highest one. So it depends how many rounds of ribosome display you want to select. Okay. So now, okay. So I just want to now to tell you that we use this protein, ABD protein, as a master scaffold to generate several types of binders. I will not time today to talk about all of them. I selected only some examples. So this is, for instance, binder recognizing P19 subunit of human IL-23 cytokine. So this is alpha subunit on this. So this is the binder, recognizing this subunit of the cytokine, and this is actually neutralizing binders. So we were looking for some blockers of interaction. Okay, So then we generated against IL-23 receptor binders blocking cytokine homology region about here. Okay, to have another set of blockers, of this receptor. Then we generated in a similar way, blockers of IL-17 receptor A, set of binders called ARS, or our first binder was targeted to human interferon gamma to identify these actually cytokine. And then we actually continued we generated a set of other targeted to human kallikreins or prostate-specific protein 94 is the prostatic cancer marker. And there is also, for instance, Shiga toxins, generated ligands. Okay, so one case, what I would like to demonstrate here, how you can use our binders is for this pro-inflammatory, starting with it's IL-23 pro-inflammatory immune pathway. Okay, one stimulating naive cells differentiate to Th cells, a cocktail of cytokines like this is the example of how they act on keratinocytes, potential treatment. It is these diseases related to this pathway. Okay, so and this is actually principle. Okay, so you have a receptor complex. One is a receptor is IL-22 receptors, and once you want to block this cytokine, this receptor can actually diminish signaling function. Then you can actually have some, like people are doing small molecule inhibitors. We are generating small proteins inhibiting the interaction also. Okay, so I just simplified, but only want to tell you that with selecting bonds, analyzed individual phases, some candidates, identify interesting ones, bonds, and then we measure in ligand tracer instrument, allowing you to measure interaction of your proteins living cells in real-time mode by fluorescent binding of your binders and detection of surface. Instrument actually accumulating fluorescence signals through the time course, giving you, like a type of curves, and you can measure and this. This is an example how we measured on nitrate samples. And this is just comparison that we generated TBD wild type, no affinity, generated affinity to that receptor. Okay, here, so you can comparison, and for instance, we also ordered synthesis of synthetic these proteins from one Chinese have these proteins. Those were our ABD types of ligands with a flag tag terminus to be able to detect it. We measured binding of these synthetic proteins to cell surfaces cells by anti-flag body. You can see that even those small synthetic peptides still bind similar way. Here you can see 5 is. This is the comparison Okay, so it's actually confirmed that these binders are. Then analyze them in many ways. I do go to just focus because we had a portfolio of binders and then we identified real inhibitors. The final experiment was done in collaboration with from the hospital. This is by Alina. Actually, they used the blood from seven healthy volunteers, then isolated peripheral blood cells, and we put them to cultivate and stimulate them for 3 days in vitro by stimulators. Just let them grow, differentiated in the presence of 23, differentiate with TH17 cells, is secreting IL-17, and we measured number of TH17 positive cells by cell sorting. And so this was the design of the, and the idea was if our binders have two expression in differentiation this type positive population primary human cells. And this is the result of this experiment. So you see that in case of minus effect, you put IL-20 only, or if you the wild type, there is no effect. But if you put no IL-20, so this is presence of no time. This is the level of number, like a natural detected number of T. Okay, there is a sample zero. Okay, you want to stimulate it more to a real. Okay, so if you put then you put IL-20 only, stimulate like this. Without this, this is the base level, and if you plate it with the no effect, so you reach about this level. If you put out binders, so you see you can really significantly suppress from this level to from only IL-20 and presence of more binders suppress modification of this cells. So this was like ex vivo confirmed effect of our binders. So we have something which could have a therapeutic effect. Okay, so then we did something similar in other projects. Is a similar idea to block time of how this IL-23 works. Okay, so this is the first kind of alpha and beta subunit, and this one unit to generate binders targeting this. Point was to select those which to block the critical part for the regulatory ligand. So did some prediction. So that binders called ILPs, they should bind in a way which is predicted to bind area which is the important for interaction with the receptor. So you see here this from the crystal structure, IL-23 know now cytokine and receptor, and there are some critical groups. If you block this part, which is important, will block this interaction. So this was like, prediction. What we are, those are examples of our binders. This is how they 19 bind. If you see where are binding site by in docking, you see mostly you see those are three most probable predicted sites for binders, and this is color coded. Most probable one is the blue one, range blue one. This is analysis of 100 binding. Most probable are those three. If you see all these binding bonds actually fit area 19 protein, which is just like in silico predicted inhibitory function. Because if those binds here, so probably should. You can see other bonds, the pattern and orientation, but still critical part of the cytokine. Okay, so then, for instance, for what these proteins can be used for. Okay, so of course we are not finding this drug candidate because they are already to do it. But, these proteins are very small, have a advantage disulfide. You can use them for excellent, tissue penetration of the antibodies. Antibodies are big, complicated. Small protein is CBD is 5 kilodaltons on, and with tax you can enlarge size. So this is a nicely produced product in bacteria. Okay. You can use it for other, for instance, studies. So this is now example of my collaboration with the Department of Biology from Josef Stefan Institute of Pennsylvania, which is headed by Let's. They are working with probiotic bacteria. They use them and they develop a secretion system how to produce and protein. Their idea is that they can develop actually another approach how to treat, for inflammatory bowel disease, Crohn's disease, because these proteins, contrary small chemical molecules, cannot apply it orally, right? Because they wouldn't go that. Because stomach. How to deliver proteins with function to treat intestinal inflammation. Okay, so they use Coccus lactis. We send our genes, then they use it to prepare construct, develop, which is the USP secretion. Now then cDNA coding our ILP or X binders. Then they use ACMA peptidoglycan anchor. So in this variant, protein is produced, transported from the lactococcal. Because there is a peptidoglycan anchor is attached and displayed on cell surface. You can detect this protein. So so question is if cells can produce our protein, if protein is produced, if is it transported and delivered. Other issue is if is it properly folded, if retains specific and if everything is fine, so then you can generate construct packing. Then you can freely secrete because Lactococcus lactis is not colonizing the strain, contrary Lactococcus bacillus types. So this is actually very nice to just put these Lactococcus lactis inside the body. They like hours, days, gut, and then they are released. So they are not dangerous to colonize intestinal wall. So it should be like safe. Okay? And during this route, okay, they are secreting. Can secrete these proteins and actually target inflammatory processes in intestine. Okay, so this is what they've done. They use bombs, divided them 9, 115, and 12. It of genetic extract with or without anchor. So they are called secreted types, and those are called display. Okay, and then they demonstrated on display assay that they actually do sorting bacteria like this, our protein variants. So there is a statistical, so proteins are there because those detected by plug, which is installed in DNF or our binder, so you can detect an anti-body. But you can do also other assays that you can use cytokine. Well, these ones are, I'm now talking about blockers of block receptor. If they block receptor, in this case you can use cytokine or in this case soluble receptor. The soluble receptor can be detected by antibodies. So you can demonstrate that once protein on the focus lattice soluble form of receptor can recognize them. So folding is proper and a specific anti. Okay, and so you can do it like this, or you can do some ELISA tests because you can measure of proteins, volume, how, and measure the decrease of free rate, etc. Depends if you use these ILPs. One set is for a receptor, design, and the other one was a cytokine. So they demonstrated that. Then I'm showing here that he did it, all these analyses to demonstrate that they are really, in case those are ILPs blockers time. So also there was a confirmed function, so showing that like this cell is capable of producing these proteins and they can be delivered to cell surface, and then yeah, used. So actually, we can summarize this part that we generated the collection of Lactococcus lactis recombinants secreting versions of Rex proteins. Versions we have like a collection those, okay, and then yeah, now are being verified in experimentally induced vitis. We got an international grant project in Sorashka and yeah, they are working with this. Milarashka is performing this in vivo experiments. They all try to intraperitoneally inject out purified proteins mostly over colon. Then they applied Lactococcus lactis normal strains, purified strains, and also they measure length. They did some section to histologists verify clinical signs of, and also they measure PCR, array of cytokines, I mean, markers of inflammation, and they got some from data, which that we really have effect of binders in this. So they even modify the methods because they use the yogurt, put the Lactococcus lactis there, and mice are eating it on daily basis. So they can plus minus control the number of Lactococcus lactis because they eat. Okay, so and they can relate it presence of effect of experimentally developed colitis. They use extra stimulated models. So this is the project is underway, just verifying the concept if we can deliver something which is therapeutically important, how to treat, intestinal inflammations. Okay, so I think conclusions are clear, and currently we have also interesting projects that are targeting binders to IL-6 receptor and is also interesting molecules, playing a role in this inflammatory process. Okay, so now I will make a break. This was like demonstration what we can do with our proteins in terms of therapeutic direction. Now I have another set results, which is another project. This project was like a demonstration how we can use our binders for research related to diagnostics. Because you can generate high affinity binders and you can ask for what they are important, how you can handle with them. So we actually started one project which is very difficult. Because, I was asked to develop protein to target insoluble fibrin. This is incredibly difficult. Because nobody in the world was successful at. The problem, they will win. So we had the project, we actually got the proposal, and it. This is like was a risky project, highly risky project. That we develop binders allowing you to identify insoluble fibrin. As a navigator for human thrombus detection and visualization. And this project was actually supported by a Czech Health Research Council, and there was a cooperation for groups. The professor is from hospital, from he was a principal investigator, and then the other groups. And we developed binders. The people from veterinary research institute in Brno were responsible for liposomes, testing there, and this head of the department from scientific help to provide MRA instrument technique to visualize. Okay. So what is this project about? Okay, so point is that there is a dream, the medical doctor. They would have something very fast detection of human thrombus, because now there is nothing such available. So, and thrombus of course causes various problems, and so there is an idea to actually to use liposomes, only approved nanoparticles by FDA, and even now EMA also used for the development of diagnostic tools. So liposomes are approved as safe. The point is now that these liposomes can be somehow targeted to reach some areas. So you need some specific binders to target them. Something you want to target cancer cells in vivo, diagnostic. You want to read something and deliver to other organs. You have to have, that will navigate your liposomes to the area, specifically where you want to reach it. So in our project was a idea. So if you use this liposome, somebody would develop targeting insoluble pin, you would be able to detect human thrombus very quickly. Okay, and this is important in some central cases because some patients who undergo some surgical operations have some expected problems, thrombus formation. So you would like to detect them more frequently than the other people. So now there is a difficult. So concept of this is called develop theranostics. Theranostics is a term meaning, okay, that you can put actually inside the liposome some drugs. You can deliver a specifically drug, so you have a therapeutic. But at the same time, you can, on the surface, you can navigate and you can use diagnostic. This is the combination theranostic, diagnostic, therapeutic, and theranostic. Okay. So an idea would be, for instance, have binders to detect thrombus. At the same time, to put inside something which would act to destroy the thrombus. And if you develop something like that, that you would see where thrombus is and how you can, destroy the thrombus as a part of. Okay, so first you have to something which navigate liposomes to these critical areas. So this is what the thrombus is. So world about 6 death over the world caused by acute myocardial infarction. In our country, which is very small, comparable to India, it's about 30,000 people are diagnosed with stroke every year, and about 6,000, high proportion, high. And some are, some other related problems, okay, and disability affect strokes. There's a problem, okay. So this is like a summary, okay, that blocking blood vessel carrying blood to the brain by a clot, okay, the brain causes a serious problems. So immediate medical treatment is so well. Therapy should be done fast as possible, of course, and now you can use a tissue plasminogen activator, which activates. Okay, can be used, but sometimes cause also problems, and they use thrombectomy removal, blocking clot. This is the standard approach for surgeons how to treat. So there is a need for direct visualization. Okay. And so this is the point. Okay, and well, this is the overview how it works. Okay, so in your blood there is a fibrinogen, the abundant protein of brains, gamma, and here it's, and actually they form disulfide bridges and start of blood clot. So there is a procedure like to first use cut peptides from A brains, then you allow to polymerize brains to develop 3 dimensions Okay, and there is other factors like okay, so from fibrinogen, which is soluble abundant protein, from fibrin, which is expression of fibrinogen. Okay, and then there is a cross-linking, forming the networks and activated platelets, also acting in this help to form the blood clot. Okay, so then okay, there is also a plasminogen activator. Activators are used to block who wants to removal or just degradation, because normally plasmin degrades with this blood clots. So during this process, there is a degradation. Then there is something like D dimers complex is formed. Now, okay, let's go idea. So problem is now that 99% surface identical. Fibrinogen, soluble fibrinogen, insoluble fibrin. This is the and this is the reason why nobody was yet able to find. Because you have to hit very very limited part of the surface, unique in insoluble fibrin over the soluble fibrinogen. This is very decent part, which is the at the end alpha ends. And there is actually a peptide which is long, as a 6 top or top, the other side of the brain. This top is soluble fibrinogen, actually not accessible. Very not accessible, and after digestion with fibrinopeptidase, polymerization now available to be recognized. Okay, so if it difficult to target something which is accessible, and then after activation, there it's very small. Okay, well, what we did actually, we looked to one paper from the one Japanese groups from Matsumura. He developed monoclonal antibody. Mouse antibody, and identify it was like okay, they identify binding these insoluble fibrin fibrils. Then they did a bit of mapping, and then they analyzed this. Okay, and during my conference in Japan, I just met one of member of his group and asked him, told him about this project, and they actually provide me. I had a tool how to monitor in our projects, how to assemble, fibrin network. So actually, our idea was first generate target, molecular target. How we can do it? Okay, okay, so let's take the. But because we know from that antibody that is formation, you cannot easily do it. We just did it anyway, but I know that the point is that it's formation. So we actually design our own protein, which is shown here. This is that we call that E top, beta top, and constructed something which is his tag as N terminal, then the sequence. Okay, then some helper protein to increase and avitat to be played and detect via. Then we did it in another way, just triple at the top. So we produce something called PEP, okay, which means triple. Okay, and then so we had a single version and triple version, and then we extracted, we call it delta protein. Delta protein is missing control. So this was our design, and we developed these proteins in bacteria and purified, and we done it. In the meantime, we generated polyclonal sera by immunization of rabbits in cooperation with some company in our country. And this is shown here that we order synthesis of the site of this terminal subate, the whole hemathion. So, and use this to immunize rabbits. We have the version so that the G epitope or G epitope, and in case of G epitope, we were successful to generate polyclonal antibodies. Okay, both rabbits produced, the rabbits were produced, okay, and generated the level of polyclonal. Did then Western blot verifying this era to the immunogen. We failed in case of the other two, so they didn't produce elicit antibodies. But we were happy with this polyclonal because polyclonal was designed against linear peptide patients. We had a broader variety of recognition. Then we analyzed our triple version or mono version of these proteins with the epitope motif. This is the delta control protein. This is the similar detection via strip pattern, so we know. Okay, and tested monoclonal antibodies from Japan group also, and they demonstrated rat clone and mouse clone. They sent us two versions. Profile is here, binding the monoclonals. Okay, and we did ELISA, and we did the same experiment with our proteins. You, the next slide. Here, this is the summary of display strategy I was before. Triple protein here, get ribosomal complexes. Actually, there is a system for ribosome display, so you see how we increase 20 the percent. Okay, and target. Okay, and we detection. This is our result. That we want to summarize that we identified several clones from several hundreds. I think we identified the 4 clones, demonstrating substantial preferential binding to fibrinogen. We are looking for. We had several candidates. Then we found that actually E7 clone is identical to 7 clones, that those two are, and so we had basically this E7 series. Okay, so by binding to fibrinogen, then tested later was eliminated. But during testing, this is ELISA referential test performed, in Institute of Hematology and Blood Diseases. So collaboration with them, this is the team of analysis for this ELISA. Okay, and then I get here out. I check the ability rate. Okay, and which is shown here. And what the point is here, by once you live here to eastern clone, so actually this clone is clones specific as we need. We did similar other analysis. Okay, we tested it. This is the same experiment. Comparison clone L. Clone, okay, referential here. So we had a control. Now, then, okay, we asked the question whether our protein ligand can be used to visualize liposomes. Okay, so this is a picture of whole blood thrombus prepared experimentally for our experiments with structure, fibrin filaments. This is the design of protein. D7 version, partner, and then okay. So we did it, the testing collaboration, and we can demonstrate the specific thrombus, I think APC within detection agent, and well, this is the only different magnitude. E negative control, which is thrombus in the presence of a type, our master version of protein. No binding, negative control. There is another control. This is so we see specific binding of thrombus via our protein. Our question was, well, for liposomes, we would need something else. We would need to attach in the oriented way our proteins to be able to visualize. For this purpose, we had to modify our proteins. Protein, our version, but we used SERP leader, okay, and terminals that we inserted flag and histag. We use histag for the possibility to immobilize protein liposomal particles with anti-NTA groups, because they produce it. NTA and NTA is a recognized histidine. So we can, via immobilization via C terminus, immobilize protein. So we designed it, we organized our protein. So in this case, they used programming enabled liposomes. Liposomes were built. Then we immobilized our protein, and we had actually two versions. And for DH, also constructing double histag version. Requirement was to have a high affinity and double histag substantially by one order of magnitude. Much heightened, stronger, binding. Yeah. So, so what you can see is, so you see cells here in both cases. Nothing, negative control ABD wild type. Okay, this is the best of cases. They are not big. This is the visualization of double hit body. Okay, so actually it works. So then we characterized, we used several other methods how to really document liposomes are made. So one method was dynamic light scattering method, allowing you to demonstrate increase of surface, which is slight shift of, because once you coat something on liposomes, the surface is bigger. Surface is bigger, so you can dynamic light scattering recognize it here. Confirm surface here. We also use micrographs here with the liposomes, visualized here by immunogold labeling, that binding thrombus particles. So it was like another confirmation that we modified. This is the confocal microscopy showing rhodamine label like one F both marks. Okay, this is the this surface of human thrombus. Okay, they actually our collaborators from the veterinary research collaborated with people from Hospital in they developed 3D model of middle cerebral artery because they art model they wanted to use practical way the photographs how it works in reality. So they actually designed a model of middle cerebral here, how they develop it, think implement and all works. Okay, but they just exactly develop what is in the human brain, okay, in this model, and they wanted to see how it works. So they use tubing, pump, okay. Then this is the thrombus here. They had a fluorescently labeled solution with our liposomes binding, and this is the result. How okay. So they used our D7 protein to visualize okay, that it there, that the liposomes were delivered there. Okay, there is occlusion, pretty tight occlusion, which is the so this is the contrast pre-labeled. So here unlabeled. So they use it to really see it or right? Okay. So this is the use of real-time monitoring of effects of various thrombolytic agents. Our point is that they would like to see also effect on thrombo degradation because they can use, for instance, plasminogen plasmine we deliver it with the liposomes are coated with our protein to be visualized. Then you can see if there is an effect of degradation at thrombus, so of that blood clot. So okay, so summary is we develop a version. Okay, we publish it in 19, but this is paper, and then we published it. It was continued, but point is that everything works nice in this model. Okay, but there is a still remaining problem. The problem is that once we really use, there is some level of cross reactivity of our protein. You generate thrombus and put S and put in 3D visualization, everything is fine. Once you go to in vivo imaging, there is still some kind of non-specificity or binding to something else. So we have to yet improve this set of useful trust agents for visualization. And we don't know actually what is the problem here. Okay, so design like pull down of a human plasma. The question was, what are these proteins? So, so we used our version with a double haze. Double haze. We put it on here in vapor, then put it to plasma. Then we take a line and wash. Then use the particles to mass analysis. Question is, which proteins will stay there bound to RD7 proteins? What are these interacting partners? Okay, and they perform the analysis here, and the number of identified peptides is 2, because the higher number of peptides you have, it's more probable interacting partner. So they identified, peptides with fibronectin, and some serum albumin, some apolipoprotein. There were some the other ones. Based on what those are. Okay, there is from one nice article. There is a set of most abundant protein in plasma. You can see it list of those. Okay, you can see that albumin. One problem. The other one is the fibronectin number 50 here. So now we have some clue, how to do, because now we want to look at the structure. Then somehow get idea, what are the critical interacting areas to be substitute protein, diminish is binding. So this is something left, then for future research. We are now at this stage. If you have a chance to, so we probably go this way, or improve me. So it actually means that you redesign some possible mutation for, let's say, in binding domain. I mean albumin in general, because our protein is albumin binding protein, so it means that there is some remaining interaction serum albumin still. Okay, so then so this is example, but otherwise this is the closest now what you can actually reach. There were people working with antibody fragments, nanobodies, also trying something, but nobody was really successful. Because one problem is between that, hematologists say, well, what you did in vitro plastic dish, this is not, fibrin. You think you generate fibrin, but this is far from reality. Okay, because if you put thrombin, to cut off peptides and polymerize, okay, it's kind of artificial type. So you can work in vitro with this, but in human body, there's many, other enzymes contributing reactions, so it be much more, modified than what you are doing. But that fact, it binds to human thrombus, which is reality. Actually makes some promise that at least you have something specific fibrin fields. Then the rest, needs to be yet done. Okay. So I don't know if you are tired or not. You have to tell art biology research. And now, if you want, I can continue with the last part, or if you want to make a break, or time is over, or tell me. Fine. So There is something about vaccines. So I just demonstrated the therapeutic and diagnostic. Okay, and so this is like what we are working with, like demonstration of. Okay, thank you for your attention. And, okay.
Ah.